feel like you could apply AI to so many different parts of what pharma companies do, right? Obviously, there's the biology discovery side, there's molecular design, there's trials. I think in many ways, the biology discovery side is probably the hardest problem you could tackle in the space. I guess you've never shied away from those types of problems, but how did you think through where to start?
Yeah, and it's a great question. And I will tell you that actually at In Citro, while we started out with a biology discovery problem, we are building an arc that goes all the way through, and we can touch on that later in the conversation. But I felt like if you look at the challenges that the industry has faced and the record and why things take so long and cost so much, it's because we make some wrong decisions often at the beginning that don't manifest until much, much later. So most drugs that fail in phase two. Some of them fail in phase three, typically because people squinted at the data in phase two and pushed forward when they probably shouldn't have. And the reason why most of those drugs failed is because they're simply going after the wrong mechanism. It's not because the pharma industry can't make molecules. They're actually pretty good at making molecules, and certainly one can accelerate that and make new kinds of molecules that people quite hadn't quite figured out how to make before. But that really wasn't the crux of the problem. The crux of the problem was we don't understand biology. We're kind of feeling our way in the dark and latching onto things that just don't make sense biologically. And so I felt like if you were really going to make a difference, that was the place to make a difference.
Yeah, and I'd love if you could kind of contextualize for our listeners like how the in vitro approach compares to maybe the traditional pharma approach, and then obviously other kind of AI drug discovery approaches like a recursion or something.
Yeah, so these are actually quite different questions. And so let me start with what the in vitro approach is, and then we can do a little bit of comparison to other entities in the space. So what we do is we try and take as data-driven and unbiased a perspective as possible. We feel like right now we're kind of feeling around with very sort of hypothesis driven, intuitive driven direction, saying, oh, this looks like a good direction. Let me explore here for a bit. Whereas what we ought to be doing is kind of rising up, collecting a whole slew of data, sort of almost from a much higher plane. And then having surveyed human biology, we can start to identify patterns and say, okay, this is a path that will take us in the right direction. So this is kind of a, if you will, an analogy. And what it translates to in practice is that as we identify certain areas of biology that we think are promising and where we feel like the technology stack that we're building has a meaningful value to contribute, we collect lots and lots and lots of data about that particular biology. And the data that we collect comes in two flavors. We print data in our internal automated wet lab capability where we generate cells that have different genetic backgrounds with different burdens of disease. We differentiate them into disease relevant cell types. So if we're working in neuronal diseases, we don't use cancer lines, we use stem cells differentiated into neurons. If we're working in liver disease, we differentiate them into hepatocytes. And then we measure them in ways that don't come in with preconceptions about measuring this is what matters to disease. We try and take as broad-based a perspective as possible. On the other side, we also bring in data from humans because ultimately, while cells are a wonderful and very intervenable model system, what we care about is curing not cells, but humans. And so we also have via partnerships data sets that we can collect that also attempt to measure human pathophysiology in a very holistic way, whether it's histopathology imaging, a modality that I worked with all the way back when I was at Stanford, MRI images, various types of omic biomarkers in blood or in tissue. And you take all these data and you feed it into the machine and you say, okay, what patterns do you see? What is the language of biology? So if you think about the large language models that are very much the talk of the day, we're building medium language models where the language is the language of cells and the language of tissues. It's not human language, it's biology language. And with that kind of way of thinking about the world, we can now ask, okay, in this language, what does healthy look like? where does sick look like? And then what is a set of interventions that might move us from one to the other? And for interventions, we look to genetics because genetics is one of the very few sources of ground truth that we have about causality in biology, where causality is very hard to come by. So that's kind of the approach that we take. And what's important is that as we take this broad landscape survey, a bunch of hypotheses emerge, and then rather than kind of closing our eyes and picking one, we say we design platforms, industrialized platforms that progress those from one step to the next. The ones that don't seem to be panning out fall by the wayside. The other ones proceed through in a wave, and that allows us to interrogate not one target at a time, but really think about what is the best way of intervening in a particular biology. So much more industrialized approach to discovery, an engineered approach, if you will, to discovery, which is really not something that exists in very many other places. So that's kind of the vision. And I did mention to you earlier, because and I think it's an important component also of the comparison, that the biology discovery, although it's been the place where we've spent the most of our effort, is only the first part of our journey. We also have an in-house capability for turning targets into drugs, into small molecule drugs. That too uses industrialized processes having to do with, you know, hundreds of millions, if not billions, of compounds that are all pooled together, attached to little DNA tags, so you could do massive chemistry experiments in a single in a single pool, if you will, and come up with a ton of chemistry data as well. And then maybe even more importantly, you mentioned clinical development as being the third phase. The work that we do with human clinical data really helps present a path forward to translating our findings into the clinic, specifically by telling us, based on data that are acquired from patients, which are the patients that are most likely to respond to a particular therapeutic intervention. And that, I think, is one of the big, I would say, misses, if you will, in the industry today, that in most therapeutic areas, cancer being somewhat of an exception, although still could be pushed forward, but certainly in other areas as well, drugs are given to an all-comers patient population. Then you have 20% that respond, 80% that don't. The ones that don't are subject to unnecessary toxicity, also not having an opportunity to take another drug that might be helpful to them because we just don't know. We don't know who should be getting what drug. And I think that's, we made such a huge step forward in oncology when we stopped treating breast cancer as one disease and we said, you know, there's BRCA breast cancer, and then there's HER2 positive breast cancer, and so on and so forth. And it created a menu of treatments that are really fit for purpose and a much, much greater therapeutic index, as it's called. Like the delta between the efficacy and the toxicity is much, much larger for these targeted treatments. Yeah. And so as we do our early biology work, it comes together with a biomarker.
Yeah.
So that's kind of the spectrum of who we are. Now, when you ask yourself, okay, so how do we compare to other companies? When I think about other smaller biotech companies, most of them end up being in one or the other of these three steps: biology discovery, target to drug, and then going and doing clinical development. The vast majority are in that middle step, target to drug. There's a good reason for that. It's well defined and easily measurable, so you know when you've succeeded. There's a few that are kind of playing an assistive role in clinical development by doing better biomarkers from whatever wearables or imaging or such. There are very few companies that span the gamut, and we're one of them. So I think that's an important differentiator. I think a second important differentiator is that we bring together both kinds of data. There's companies that do discovery using primarily cell-based functional genomic approaches. There's companies that do discovery based primarily on human clinical data and looking mostly for genetic or genomic correlates. There are very few companies that really bring the two together, and we think that they really synergize with each other in a way where the whole is considerably larger than the sum of the parts. And then I would say the third differentiator is just the scale of automation and data creation that we've put together, which I don't want to say is unique. I mean, certainly you mentioned recursion. It's probably the one that has made the biggest investment into the creation of data at scale. They started before us, and they certainly have done an incredible job of putting in place that type of automated data creation. So we are in that same bucket, if you will, but they only do the cell-based stuff
Right.
and focus on the early discovery. Now they're starting to move into molecular generation, whereas we're kind of, as I said, by bringing the human data component into this, we think it gives us the ability to really bridge the gap between cell biology and translational medicine.